Right, thank you and good afternoon. I'm Steve Ellison at LGC Teddington, National Measurement Laboratory for Chemistry in the UK. Today I'm going to be talking about some of the work that we've done on producing meat species reference materials. I'm going to talk a bit about why we do that, and then I'm going to talk at a bit more length about how we go about value assigning pure species and mixture materials for meat species. Part of the reason for doing that today is that the basis for characterization is essentially provenance, that is, evidence of the origin of the material. So this forms an example of a provenance value assignment, although we do, of course, provide some checks along the way. And I'll talk briefly about stability and homogeneity, which is somewhat less important for single species meat materials. So the background to this goes back to 2013, and many of you will remember that in 2013, Ireland and the UK started to find horse meat in what should have been beef in a number of food products. Now, horse meat isn't unsafe of itself. It's just that in the UK, it's very unusual. It's not supposed to be there. And there's significant concern that if horse meat is finding its way into the supply chain for beef products, that the horse meat isn't necessarily edible when it gets there. So the incident took off in quite a big way, you can see from the headlines, and generated quite a lot of government activity. And part of the government activity was to refer to LGC, because one of the things that was urgently needed was a lot of testing. One of the things that wasn't available at the time was a set of positive and negative controls for horse meat in beef. So the Food Standards Agency in the UK needed someone to produce some reference materials pretty quickly for essentially qualitative tests. We also needed pure materials because those can be used to calibrate, for example, PCR. So we ended up working with the FSA to produce a set of meat species reference materials. All of the single species meat materials fall within our 17034 scope, so they would now fall under ISO 33406. We produced a number of different kinds of materials. Some were single species, some were mixtures, arranged at various times in the last few years, up to 7 different meat species and 6 mixtures. And at the moment, or rather the most recent production of mixtures was at approximately 1% contamination, which is often regarded as if you like, an upper limit for adventitious contamination. And as I've already said, the primary characterization is by provenance. You can see there, we start with a cow and we end up with a reference material, and if we know that that's what we started with, it's pretty clear that what's going to be in the jars is beef. So next question is how we go about ensuring that. And I'm going to talk through an example, which is a relatively recent certification of 2 materials. You can see the batch numbers there. The beef is batch 8 and the pork is batch 7. So we've produced quite a number of batches over the years since 2013. Now, let me start by talking a little bit about the characterization. The point of this is that we value assign by evidence of origin primarily. It's not the only evidence we've got, and I'll talk in detail about that. But the idea is that you start with an animal, on a farm, you obtain meat from the animal, process it, put it into jars, and as I said before, what's in those jars should be the same species as the animal you start with. There's very little doubt about the species under these circumstances. We're not relying on testing which could give different results. Most people know what one of these animals is. In practice, part of that process is in our control, so we can keep quite tight controls on that. Part of it is controlled by the supplier, and of course we're in an environment where we don't always trust the suppliers of beef. However, we can do something about that. So one way of looking at how we're going to manage this is to look at some of the risks involved in a characterisation of this kind. So the bit that's not in our control is the supplier evidence. The first defence we've got there is to find a reputable supplier with good documentation. We have tended to use organic suppliers because they're already used to providing thorough documentation to maintain their organic certifications. So that's part of our defence against supplier malice, if you like, supplier fraud. Very unlikely in this circumstance, of course. The other things that can go wrong are that the supplier can put the wrong meat joint and mislabel it in the bag, as it were. And in principle, sample can get confused in the laboratory once it's arrived. The main defence there is that we add a number of confirmatory checks, particularly based on next-gen sequencing, which I'll talk briefly about later. The next major risk is that somewhere in the lab we cross-contaminate the meat, because one of the things we're trying to produce, for example, is a beef material that's known to contain no horse meat. This is a negative control, and we provide positive controls with mixtures, but we need a negative control to begin with. And testing labs need negative controls that don't contain a suspect contaminating species. So we want to be pretty careful about cross-contamination with other species, particularly since in principle we're handling several species. So we've got our own contamination control procedures in the lab, and the other thing we do is some further analytical checks on cross-contamination. So if we go through the process, we start with acquisition. We've got pre-approved suppliers for all of the species that we're interested in. In some cases we've audited them. They all provide documentation of what we've ordered and what we've got, and we end up with effectively relatively small quantities of meat, because we'll see later that we generally produce these in relatively small batches. They don't have a long shelf life, which we'll get onto. Once we've got our material, we're talking about production. Essentially, you're starting with a large piece of meat and ending up with a small amount in a bottle. Most of that is fairly routine. The reason that looks fairly clean is that one of the first things that's done is to take the surface material off, and that removes connective tissue and fat. It also takes off any surface that might have come into contact with something else at the supplier end. And once we've done that, we effectively reduce it down to ground beef, mix it thoroughly, and place it in whatever container we want. We use 15mm amber glass. During that process, we don't want anything else to touch any of the meat on the way, particularly at these stages where there's no protection. So what we have contamination control protocols. We never have the same material in the lab at the same time. It's trimmed, as I've said before, to minimise contamination. We've got completely different sets of equipment for each different species. So none of the equipment used for beef is ever used for another species, and vice versa. Work surfaces, equipment, utensils cleaned before and after use. Fairly standard cleaning procedure, but thorough, so that there's essentially no chance of things like nucleic acid contamination, because we want our materials to respond to DNA tests. We expect DNA tests for some of them. And lab staff use PPE, clean lab coats, disposable aprons, and so on. So essentially the same sort of near sterile handling that you'd expect in a good food production area. In principle, that means that we are pretty sure that we haven't contaminated the material. Once we've got the material packaged, we start on our confirmatory analysis. The first one is to make sure that we've actually got the material we think we have, the species we think we have. We do that by next-gen sequencing at one of our partner laboratories inside in the LGC company as a whole. So there's a LGC Genomics in Berlin, do nucleotide sequencing, and you can see there it uses mitochondrial 12S and cytochrome B, which is fairly standard for species identification. So we end up with results from them which tell us which species they think we've got, and in fact which haplogroup. And we've never had a surprise, but in this particular example where we're looking at a beef and a pork material, everything matched as we expected, so we were pretty sure that we've been sent the correct species. At the same time, we're doing cross-contamination checks. There are two that we do. One is DNA based on PCR. One is ELISA based, so we're checking both the DNA response and the protein test kit response. They both use duplicate analysis of two separate units. It's not a large test, it's a check, but it's sufficient to check for, if you like, homogeneous contamination of the ground material. They don't have particularly low detection limits, some of them. 0.1% for horse meat is pretty good, because we're looking typically to detect 1%, 1% for some of the others. For ELISA, the kits that we're currently using have detection limits of around 1%. They don't distinguish between sheep and goat or chicken and turkey, and that's important sometimes when we're looking at some of our poultry materials. For this particular example, it's quite sufficient to tell us whether there's one of those species present. Typical results are usually reassuring. You can see that we've tested our beef and pork materials, two samples in duplicate for 7 different species, and what we get is positives just for the species we expect. So in this particular case, using the PCR test, we're sure that we only got detectable DNA, certainly at the 1% level. The ELISA test, we get pretty much the same results. When we're testing beef, the species detected is cattle. It's bovine, and for pork, it's porcine, so it's pig. We don't get any evidence of cross-contamination there. Packaging and storage is, as I said, amber glass bottles. These particular materials are sold in 10-gram units for the pure materials. And as I said, we don't produce a great number at a time. Where we've got additional bulk material, we do use that for producing mixtures later. And our current storage is at -20 plus or minus 10. I'm going to talk a little bit about stability assessment, because we have done some in the past, but our stability approach is somewhat different for new batches now. So the thing about stability of a meat species is that the property of interest, the species, is not going to change. What can change is the response to our DNA and protein test kits. So we've primarily relied on checks on past batches to cover that. We've also done checks to make sure that we can still identify the species after storage for 2 years. The other thing that we've done, because the materials are frozen and that can denature DNA or proteins, we've done some freeze-thaw cycle checks. We're going to freeze it once to put it into storage. It's quite likely that the user is going to put it into cold storage as well, so we've checked to make sure that 2 freeze-thaw cycles don't materially impact the nucleic acid response or the protein response. I said I'd talk a little bit about the long-term stability checks, which we've done quantitatively, and if you're familiar with ISO 33406, which is what we're talking about today in part, it does say if you can do something quantitative, please do, because you get more information. And you can see here that while we get reliable detection of pork or horse or turkey, and these are 1% materials in either beef or chicken. So you can detect them as positive or negative, but what we are seeing is a change in the cycle time, and the cycle time goes up as the amount of amplifiable DNA goes down. So if we have a quick look, you can see there's a very noticeable gradient for pork and horse DNA, somewhat less so for turkey. The practical upshot of that is that we don't think the DNA response is sufficiently stable over time to use these quantitatively or to keep them for any length of time at -20. We can also look at short-term stability, which is useful for transportation. We have done that on previous batches. This is an example of 1% horse meat in beef. Just having a look at it, the structure of the experiment is that we've got 3 different times in weeks. So we've got 0, 2 and 4 weeks. So we've exposed it for up to a month and examined it 3 times. And we've got 5 temperatures there going from -20 up to +60. These are all ELISA determinations. And what's happened here is that for each of these squares, we've measured 3 units, or rather we've tested 3 units in duplicate after exposure. The solid colour here is telling you how many of the individual observations were positive. In this case, you can see that they're all 100% positive, which is reassuring, except at 60 degrees, where effectively the material doesn't survive 60 degrees for probably more than a week, because we're just seeing nothing at all after 2 weeks at 60. Essentially, we've denatured the protein there beyond detection. So while we could ship at ambient if we were confident the temperature would stay below, let's say, 20, there's obviously a small risk that if it gets to higher temperatures, we might end up with some degradation of the material. Going on to homogeneity, we can do similar types of experiment. So again, this one's pork in beef, the 1% material, and we're looking at whether we can detect it or not. We're not measuring it quantitatively because we're not expecting a perfectly uniform distribution of pork in beef anyway. What we are expecting is enough to detect reliably. So we've looked at 18 units of our reference material, which, if you remember, for the batch size we're looking at is quite big. I've tested them by ELISA, and we've run 5 replicates per unit per reference material unit. And a similar picture there. You've got your 18 units along here. None detects. 0% none detects for all of them, and for all of the materials we're seeing 100% positive. If you're interested in what that's telling you about the possibility of defective units, it actually isn't very informative. If we calculate the confidence interval for that, the lower 95% proportion, lower 95% limit for proportion positive, we can actually only say from this that we're pretty sure that more than 87% of our materials are satisfactory, and that's because it takes an awful lot of individual units to get this number up around the 95%. If you want to detect 1% of failures, you need to run 300 units, and that is simply unrealistic for the type of analysis we're doing here. So we've got fairly good evidence that there's no variability across units in our test kits at the moment. Having said that, our current approach to homogeneity and stability is that we no longer do homogeneity or stability for new batches. All of the material is one species. We know that. We've got no evidence of contamination. So we know that everything that goes into bottles will test positive for the right species. And that's backed up thoroughly by confirmatory checks and contamination checks. So we're very confident of the value. The mixture materials are individually weighed from homogenate, or at least were early on. So we give a nominal level, but we give any mass that we give, any proportion we give is specific to the individual unit. So we don't do homogeneity testing for those. Stability, because of the evidence of DNA degradation over time, we simply don't retain the material past 12 months, and that's why we make small batches. So we've historically made one batch a year and discarded anything that was not sold. So we don't do stability testing for new batches. We have done some long-term monitoring, and that's what I've shown you previously. Certificates for these, very simple. It just says ground raw beef. All we're saying is it's the assigned species. We don't have a statement of confidence there. If you turn over the page, though, you'd find that there's considerable information about how we've confirmed that identity. For mixtures, we give a weight of raw material and the proportion that's actually been weighed into the sample. We don't put uncertainties on that because these are sold essentially as nominal values. The practical upshot of all that is that we've got a range of materials that we've characterised just for species. It's a nominal property, so it doesn't have a number. The primary characterisation is via provenance, the evidence of origin, with confirmatory speciation. That's backed up by, or ensured, if you like, by pretty careful contamination control in the lab, plus our confirmatory checks are for contamination. Our assessment of homogeneity and stability is primarily previous experience, so we don't need to do homogeneity and stability for new batches. And on the plus side, it was a lot of work for the reference material team, but they managed to get suitable materials out in record time to support an awful lot of UK testing laboratories trying very hard to find out what foods in the UK were risking contamination. And having said it's been a busy time for the reference material team, just a few acknowledgements. The initial project setup was funded by the Food Standards Agency in the UK. The reference materials team that are responsible for coordinating all this work is Jill Holcomb, the team leader, Laura and Marina, who cover the individual projects, and the other work that I've talked about, LGC Genomics in Berlin, our molecular and cell biology team in Teddington, and our government chemist team in Teddington for doing the different analytical work that I've talked about. And that's it for today, so thank you very much, and questions welcome.